Hi guys, we're students at the University of Oklahoma and we'll be talking about how we can use microorganisms, such as E. coli, as factories to make useful products. Specifically, we'll be talking about insulin. Frederick Banting and Charles Best discovered insulin in 1921. Insulin is a protein hormone which regulates fat and carbohydrate metabolism. And when your body can't produce enough insulin, you develop diabetes. So in order for your metabolism to function properly, you need insulin replacements. This replacement insulin used to be developed from bovine and porcine, cow and pig pancreases up until 1980. Insulin isolated from animals differs from human insulin, even if only slightly. For example, cows have a 3 amino acid difference and pigs have a 1 amino acid difference from the 51 amino acids found in human insulin. Furthermore, it takes 2 tons of animal parts to make 8 ounces of usable insulin. So in 1978, scientists at Eli Lilly and Genentech found a way to use E. coli to make insulin. This insulin was the first recombinant protein approved for human use by the FDA in 1982.
Diabetes often occurs at old age, and as people live longer, the overall need for insulin increases. The reason why E. coli is chosen is because it has a high growth rate, it is easy to use, and it is very cost effective. So it's not a surprise that 24% of biopharmaceuticals use E. coli. Also, by using E. coli, we can modify and improve insulin to make it more effective, more stable, and cause less of an immune response. Using E. coli to make insulin generally consists of 3 parts: gene transfer, growth, and purification. The first step is to introduce the human proinsulin gene into E. coli. To do this, the mRNA for proinsulin is isolated from the beta cells in human pancreas. The reason that mRNA is used is because it should already have the codons for the introns removed, so it should only include the coding parts of the gene. Reverse transcriptase is used to make complementary DNA, cDNA, and then DNA polymerase is used to make the cDNA double-stranded. Now this DNA clone for proinsulin has to be inserted into the E. coli. To do this, we use plasmids, small circular strands of DNA which can be transferred to bacteria. A plasmid such as PBR322 is used as a cloning vector, as a vehicle to transfer DNA. First, a restriction enzyme, such as EcoR1, is used to cut isolated plasmids at a specific site. Then the DNA for proinsulin is stitched into the plasmid using DNA ligase. The plasmids are introduced into E. coli, which absorbs them in a process called transformation. In order to speed up the process, CaCl2 and heat shock is applied to the E. coli. The second step is growth, where the E. coli are grown in progressively larger and larger containers, starting with petri dishes filled with growth medium. In these dishes, repressors are added to prevent the E. coli from producing insulin, and ampicillin, an antibiotic which interferes with peptidoglycan synthesis, is added to prevent the growth of E. coli, which did not absorb the plasmid. The reason this works is because the PBR322 plasmid contains genes which offer resistance to ampicillin. Therefore, those E. coli which did not absorb the plasmids will be susceptible to ampicillin. After enough growth, the E. coli is transferred to a bioreactor, a fermenter. An activator for proinsulin expression is added, and the temperature, pH, and oxygen supply of the reactor is monitored. The third and final step of the process is purification. So the proinsulin molecules that the E. coli make are collected within the bacteria's inclusion bodies, which are essentially protein aggregates. In order to recover these proteins, the bacterial cells have to be lysed, broken, using processes such as sonication. The cell contents are centrifuged and filtered for initial recovery, and finally chromatography is used to obtain a purity of 99% or higher. Chromatography separates a species in a liquid based on their affinity to a solid. It is a highly effective technique. Since E. coli have different post-translational modifications compared to human cells, we have to modify the proinsulin further to get the final properly folded insulin hormone. First, the connecting peptide, C-peptide, needs to be removed through proteolytic cleavage. Second, 3 disulfide bonds need to be introduced, which are not effectively produced using E. coli's own mechanisms. After these changes are made, a final insulin crystal is made, ready for use.